So we're going to kick things off in this chapter by studying the cell cycle. The cell cycle is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughter cells divide. Just before the cell begins to divide, it's going to undergo a series of transitions. First, it's going to grow bigger. The number of organelles is going to double. Cell is going to go DNA replication, which leads to doubling of the genetic component as well. This first stage of doubling and growing is the first of two major stages of the cell cycle. So the cell cycle is a predictable set of events that the cell goes through from its origin to its own division, from its origin to its division into two daughter cells. The two major phases are going to start off with interphase. Interphase is when we're going to grow and double and perform all of our normal functions as a cell. The second stage is going to be called the mitotic stage, also known as cellular division. This phase includes mitosis and cytokinesis. Here is a diagram of that cellular division. We're going to see that in this phase, this gray phase over here, this is what's known as interphase. The cell is going to spend most of its time in interphase. Then we're going to go into cellular division. Cellular division is going to occur in this phase. We're going to undergo mitosis and cytokinesis, and we're going to end up with two cells where we initially had one. This process of mitosis can be broken down into further phases: prophase, metaphase, anaphase, and telophase, and we're going to spend some time going through each one of these phases, starting off with interphase. Interphase is going to be the period of growth and preparation for cellular division. It's not a resting period. When I was in school, I was taught that this is the resting period. This is the relaxing period. No, it is not. There is a lot happening in this so-called resting period. This is where we are going to be undergoing a DNA replication, which is a huge process in and of itself. We're going to be performing all of our normal functions. So if it's a liver cell, it's going to be detoxifying the blood. If it's a pancreas cell, it's going to be secreting hormones. So during interphase, all of our normal functions still have to occur while the cell is doubling not only its DNA, but also doubling in size. There are three distinct phases for interphase. They are known as the G1 phase, the S phase, and the G2 phase, and we're going to cover each one of those. So in interphase, G1 is you can think of G standing for growth. This is our first growth phase, the first gap phase. This is going to be a time of major growth before DNA synthesis begins. At this point, the chromosomes are going to consist of a strand of DNA and proteins. And so we are going to grow up the cell to help for the process of DNA synthesis. DNA synthesis, which we will see, is a very complex process. And it's going to require a lot of proteins, a lot of factors. And so during G1, we are assembling the much needed proteins to undergo our synthesis phase, our S phase. The next phase is going to be our S phase. S phase is short for, or you can think of it as it's standing for, DNA synthesis phase. In this stage, DNA is going to be replicated. We're going to make two copies of our DNA called chromatids. They're going to remain attached at the centromere. At this point, the DNA is still loosey-goosey. We still have a plate of spaghetti. We haven't condensed down to raviolis yet. So at this point, we have loose DNA, but it's all replicated. The two attached chromatids are going to be genetically identical and are called sister chromatids while they are still attached. Growth is also going to continue on through this phase. The next phase is going to be the G2 stage, or the second gap phase. This is going to be the period after DNA synthesis and before mitosis begins. Again, this is not a resting phase. Yes, we have already doubled our DNA, but we still need to double the rest of the cell. The cell does need to completely double in size, and all of its organelles are going to have to be duplicated. So growth is continuing on through this phase. What we're going to see is over the process of the next couple of phases, our DNA is going to condense down. When our DNA condenses down, we go from having these great big long strands of spaghetti, and here they're shown kind of in the relative size of a chromosome. They're not. Think of this as a crazy long strand that extends way beyond, extends 3 feet beyond my computer screen in either direction. A very long spaghetti strand. During the process of mitosis, that DNA is going to condense down. Those strands of DNA are going to wind around histones. Those histones are going to coil around each other. They're then going to zigzag back and forth into radial loops. Those radial loops are then going to be found wrapped around each other again and found inside of these chromosomes. So these are highly, highly condensed structures. Highly condensed structures when we undergo the process of cellular division. So next up, we're going to talk about division of the nucleus and the cytoplasm. This is going to consist of 2 processes. The first is mitosis. Mitosis is division of the nucleus, and cytokinesis, which is going to be division of the cytoplasm. We are going to see that these processes will occur simultaneously, but since they have different end goals, we'll talk about them as separate processes. So here is sort of a very short overview of mitosis. We're going to see our DNA condensed, lined up down the center, separated, and 2 new cells are going to form. Because we're essentially taking 1 cell and cutting it in half into making 2 more cells, this first cell needs to be very big. This is why we have so much time in Interphase devoted to just growing the size of the cell. Because if we were going to take a single cell and cut it in half, and then cut it in half, and then cut it in half, and then cut it in half, with no growing in between, those cells are going to get infinitesimally small and useless. So the cells always have to grow before they can undergo division. All right, so we're going to talk about this process of the division of the nucleus and the cytoplasm. We're going to see that in the developing embryo and fetus, body cells are going to divide continually. In small children, we're going to see body cells divide for growth, but also repair. We're going to start to see, especially children, they tend to be very clumsy, lots of scraped knees, lots of broken bones. So we're going to see lots of growth and repair in small children. In adults, we're going to find that some cells are going to lose the ability to divide, so specifically your neuron cells. So if you've ever heard that before, that, once you lose a brain cell, you lose it forever. Yeah, your neurons are going to lose their ability to divide and replace. Not all, but most of them. Some cells are going to stop dividing, but they do retain the ability to divide should the need arise. So for example, your liver cells. Your liver does not just keep growing. If your liver kept growing and never stopped growing, by adulthood you would be mostly liver. Not the case. Your liver does stop growing at a certain point and only continues to grow if needed. So if you were to lose a part of your liver, if you were to donate a part of your liver, your body could regenerate that liver. And then there are some cells that are going to actively continually divide throughout life. For example, a lot of epithelial cells. A lot of epithelial cells are going to be constantly being replaced, constantly undergoing mitotic division throughout your full adult life. Mitosis is going to be the process of specific nuclear division, and we will cover that in our next section.